Hi boys, this is your biology teacher Shakeel Ahmad, and today we will discuss the function of a Lac Operon. Lac Operon is actually concerned with the control of the gene expression. We have a number of genes present in our cells, and these genes they code for the proteins. Some of the proteins are being they are constantly required by our body, like the biochemical processes such as respiration, and these proteins will be produced by these genes all the time. But there are some genes that code for the proteins that are not needed all the time. So these genes are switched off when their proteins are not needed. The best example of this control of switching on and off a gene is the operon. An operon is a group of genes that are transcribed at the same time. They usually control an important biochemical process, and these operons are only present in the prokaryotes. The Lac operon is the best example of the gene expression control. A Lac operon consists of three genes, each involved in the processing of the sugar lactose. The most important of the three genes is the gene that codes for the enzyme beta galactosidase, the gene Z. This enzyme hydrolyzes lactose into glucose and galactose. Now, the bacterial cells, for example, the E. coli, they have preference to glucose or they prefer to use glucose than lactose. So if both glucose and lactose are present in the medium, they will not waste energy in hydrolyzing lactose, and they will prefer to use glucose. So a bacterial cell may be confronted with any of the four situations. One, when glucose is present and lactose is absent in the medium or in the cell. So at that time they don't need to produce beta galactosidase because glucose is already present. When the second situation is when glucose is present and the lactose is also present. Again, the bacterial cells, they don't need to produce beta galactosidase because glucose is already present. Why would they lose energy in hydrolyzing lactose? Third, when glucose is absent and lactose is also absent, again this time the beta galactosidase will not be produced because its substrate lactose is not present in the medium. So the fourth situation, when glucose is absent and lactose is present inside the bacterial cell, the enzyme beta galactosidase will be produced by it. So when lactose is absent, that time how are the genes switched off? There is a repressor protein that's being produced by a regulator gene, and this repressor protein binds to the operator site. It keeps the operator site blocked so that RNA polymerase cannot bind to the operator site. When RNA polymerase is unable to bind to the operator site, the three genes will be switched off. When lactose is present in the medium, a small amount of the allolactose will bind to the repressor protein, which is actually occupying the operator site. This binding of the lactose will cause a change in its structure. That's what we call the conformational change, and it will leave the binding site. So the binding site will be free for RNA polymerase to bind to it, and the gene expression will start. When lactose is present and glucose is also present in the medium, the lactose will bind to the repressor protein, and repressor protein will leave the binding site or the operator site for RNA polymerase to bind to it. But the RNA polymerase at that time is unstable and it keeps falling off from the operator site. So in case when there is glucose not present in the cell and lactose is present, that time another protein is produced in the cell. That is known as the activator protein. This protein actually stabilizes the RNA polymerase when it binds to the operator site. So the repressor protein has already been changed and it has left the binding site by the binding of the lactose molecule to it. Then the RNA polymerase, it binds to the operator site, and at the same time the activator protein actually it steadies the RNA polymerase and it keeps it bound to the operator site, and the three genes LAC Z, LAC Y and LAC A, they will be turned on and they will express themselves to produce the enzymes. Now this process we can understand by an animation that is following. Yes boys, now we will understand the working of a Lac Operon by this animation more clearly. So we have the Lac I promoter, and this is the Lac I gene which codes for the repressor protein. We have the Lac Z which codes for the enzyme beta-galactosidase, and the Lac promoter is over here which should go to the promoter site, and this is the operator site where the RNA polymerase binds. So you can see it over here. When lactose is not present in the medium, the Lac I promoter site has initiated and Lac I is turned on, and it is transcribing the messenger RNA for producing the repressor protein. The repressor protein will now be translated. It's formed over here, and it binds to the operator site of the Lac Operon. Now, as it has occupied the operator site, the RNA polymerase is not able to bind to the operator site, so Lac Z is not able to express itself. Now let's add lactose from here. When the lactose is present inside the cell, now this lactose binds to the receptor protein. You can see it here. It has caused a change in its shape and it has left the binding site. So RNA polymerase binds to the operator site. It's able to, and now Lac Z is expressing itself to produce the messenger RNA. The messenger RNA will now translate to produce the enzyme beta-galactosidase, which breaks this lactose into glucose and galactose. So as long as we have lactose present in the medium, the repressor protein will not bind, or it will leave the operator site free, and Lac Z will be able to express itself. So if we can, these are actually the symbols that we are using for different parts of this Lac Operon over here. Now let's understand how does the lactose transport into the bacterial cell from the medium take place. Now here we can see we have the Lac I promoter site, so it should go to its promoter site. We have Lac I gene over here. Lac Z should go to its site, and Lac Y, which actually produces the enzyme permease, which allows the movement of this lactose from the medium into the bacterial cell. Then we have, this is the operator site, and this one is the Lac promoter. Now as we don't have lactose in the medium right now, Lac I has expressed itself, so repressor protein is formed. Repressor protein has is going to bind to the operator site. Now it's blocked, so RNA polymerase cannot bind to the operator site, so neither of these genes, Lac Z or Lac Y, will be able to express themselves. Now let's say there is lactose present in the medium. When the lactose is present in the medium, we are adding more and more lactose to it. Now we'll see the molecule of the allolactase will bind to the receptor protein, and now this is Lac I. It has produced the messenger RNA for producing the receptor protein. Now we can see it here. Still the repressor protein has kept the site occupied. Now it has left it. Repressor protein, it has, because there is no lactose present in the cell, so it is occupying the operator site. Now we'll wait for the lactose, for the repressor protein to leave the binding site, so that Lac Y can transcribe the protein which permeates, which will allow the movement of lactose into the cell. Now we can see it over here. Now the binding site is free. The RNA polymerase has bound to the operator site. Lac Z is not expressing itself first. It will be Lac Y that will express itself. Now Lac Z has also expressed, and Lac Y is going to express itself. It's Lac Y producing the messenger RNA, and this messenger RNA will now translate to form the permease protein. Now this is beta-galactosidase first from lactase Lac Z. Now this is permease protein, which will fit itself into the membrane as a transmembrane protein, and the lactose molecules will move into the bacterial cell. And as they move into the bacterial cell, we have already the beta enzyme beta galactosidase is present, which will break the lactose into glucose and galactose. So the presence of or the expression of both these genes, LAC-Z and LAC-Y, is very important, because LAC-Y transcribes for the protein permease, which allows the movement of lactose into the bacterial cell. I hope these two animations help us to understand the process of Lac Operon, or the control of the gene expression, more clearly over here. Thank you very much.